We piloted the use of liquid biopsies to support a diagnosis of patients with suspected cancers across multiple tumor types, but this included pancreatic and biliary tract cancer. We know that these particular tumors are quite difficult to diagnose given their deep location in the body. They often require invasive diagnostics such as endoscopies and also repeated investigations as well. And this results in delays in diagnosis and starting treatment. And this was this was obvious during the COVID-19 pandemic when worldwide there was a reduction in invasive diagnostic procedures. So we piloted the uses of liquid biopsies to support a diagnosis in these tumours, and we did need a non-invasive way of diagnosing these tumours. So liquid biopsies or ctDNA is DNA that's tumour-derived. It's released from the tumour into the blood, and it can be detected and analysed through a liquid biopsy. In this study, in the PREVAIL study, we enrolled patients with suspected malignancy, so radiologically suspicious disease, who had not had a confirmed histological diagnosis. We recruited 16 patients per cohort, including in the biliary tract and the pancreatic cancer cohorts, and we analysed and collected ctDNA using a custom 59 gene panel at the Institute of Cancer Research, our in-house assay. Subsequently, all results were interpreted used through a molecular tumour board with genomic specialists' input. We found that in our cohort that we had a detection rate in the pancreatic cohort of 69% and in the biliary tract cohort of 56%. And remember, some of these patients didn't have malignancy, but radiologically suspicious disease. So this is what we were expecting. Some patients subsequently underwent a biopsy, and that was 23 patients across the two cohorts, and we were able to do a sensitivity and specificity analysis. We actually showed relatively good high sensitivity and specificity when we used a liquid biopsy or ctDNA in the diagnosis of pancreatic and biliary tract cancer. The sensitivity was 80% for the pancreatic cancer cohort and specificity 100%. And in the biliary tract cohort, the sensitivity was 100% and specificity 75%. Interestingly, we did detect something called a false positive result in the biliary tract cohort, which did actually reduce the specificity. However, that patient was subsequently diagnosed with an esophageal cancer, not a biliary tract cancer, and that was why the patient had a detectable ctDNA. So we found from our study that liquid biopsies and ctDNA is a non-invasive tool which can be used to support a diagnosis of cancer in patients with suspected pancreatic and biliary tract cancer. We found good sensitivity and specificity when ctDNA was used as a diagnostic tool in these tumours. And we thought that in these patients who are unable to go invasive biopsies due to, say, comorbidities or in difficult to access tumours, or those requiring repeated biopsies and investigations, we could use ctDNA to accelerate that diagnosis. The approach that we piloted may in fact improve diagnostic efficiencies and add resilience to the current pathway, which is currently reliant on invasive diagnostics to diagnose these difficult to diagnose tumours.